Imagine a microscopic city where bacteria cooperate, build walls, and create channels for nutrients. Today we'll explore the step mechanisms that allow these tiny organisms to stick together and form resilient biofilms. Bacteria in the wild often exist as free-floating or planktonic cells. When conditions favor attachment, these cells begin to adhere to a nearby surface. The first contact is mediated by weak electrostatic forces and hydrophobic interactions. Once attached, bacteria produce an extracellular polymeric substance, or EPS, that acts like glue. EPS is composed of polysaccharides, proteins, lipids, and DNA fragments that bind cells together. This matrix creates a protective environment that shields the community from shear forces and chemicals. As more cells join, the biofilm expands into a layered, structured architecture. The resulting biofilm can be found on natural surfaces like rocks or engineered ones such as pipelines. In ecosystems, biofilms contribute to nutrient cycling and habitat formation for other organisms. In industry, they can cause corrosion, clogging, and reduced efficiency in water systems. Understanding the initial adhesion steps is essential for managing both beneficial and harmful biofilm growth. Pause 0.8. Bacteria first encounter a surface while swimming in liquid. Their outer membranes carry small molecules that can sense nearby materials. When the cell approaches, weak van der Waals forces begin to pull it closer. These forces arise from temporary dipoles between atoms on the bacterial surface and the slide. Simultaneously, hydrophobic patches on the membrane see contact with nonpolar regions of the glass. The attraction reduces the energy barrier for attachment, allowing the cell to settle. Surface roughness can trap bacteria in micropockets, increasing local concentration. Once adhered, the bacterium secretes a thin matrix that anchors it further. This initial adhesion is reversible until the extracellular polymeric substances harden. The process sets the stage for subsequent layers of cells to join and mature. Pause 0.8. Bacteria begin by secreting small sugar chains called polysaccharides into the surrounding fluid. These polysaccharide strands quickly spread and stick to neighboring cells. At the same time, protein molecules are released and weave through the sugar mesh. The proteins act like connectors, binding both sugars and other proteins together. DNA fragments from lysed cells also enter the mix. They become extracellular DNA or eDNA. eDNA provides a rigid scaffold that helps hold the whole structure in place. Lipid molecules are secreted as tiny droplets that embed within the mesh. The combination of sugars, proteins, DNA and lipids forms a dense polymer network. This network traps water and nutrients, creating a protective environment for the bacteria. Over time, the matrix thickens and hardens, sealing the bacterial community together. The result is a stable biofilm that resists removal and external stresses. Pause 0.8. Microcolonies begin as small clusters of bacteria attached to a surface. As they divide, cells secrete extracellular polymeric substance that glues them together. The EPS forms a matrix that traps water and nutrients around the colony. Within this matrix, some cells become specialized for nutrient transport. These specialized cells create micro-Uros channels by secreting enzymes that dissolve local EPS. Channels allow water to flow deeper into the biofilm, carrying oxygen and food molecules. The flow establishes a gradient where outer layers receive more nutrients than inner ones. Cells in lower Uros nutrient zones shift metabolism to conserve energy and produce more EPS. This dynamic balance builds a layered architecture with dense cores and porous peripheries. Over time, the biofilm thickens, forming distinct strata that resemble miniature ecosystems. The mature structure supports longer Uros term survival by protecting cells from environmental stress. Pause 0.8. When a biofilm reaches maturity, bacterial cells begin to sense their environment through chemical signals. Quorum sensing molecules accumulate in the surrounding fluid and trigger intracellular pathways that lower adhesion. A key regulator, cyclic di-GMP, drops in concentration, prompting cells to produce enzymes. These enzymes include proteases and denases that break down the extracellular polymeric matrix. As the matrix thins, surface tension forces become weaker and cells can detach more easily. Detached bacteria then regain motility by re-expressing flagellar genes, allowing them to swim away. The dispersal process is reversible. Planktonic cells can later encounter surfaces and initiate new biofilms. Environmental cues such as nutrient depletion or pH shifts accelerate the signaling cascade that leads to detachment. Mathematical models predict that a threshold concentration of dispersal enzymes is required for successful biofilm breakdown. Understanding these biochemical switches offers insight into how bacterial communities adapt to changing habitats. Pause 0.8. Shear stress from fluid flow exerts a mechanical force on bacterial colonies. When the force is high, cells are pulled apart and biofilm density decreases. In stagnant environments, the lack of shear allows cells to remain close together. Nutrient availability also dictates how much extracellular polymeric substance a cell can produce. Rich media supply precursors for polysaccharide synthesis, leading to thicker biofilms. Conversely, nutrient limitation forces bacteria to conserve resources and reduce EPS production. pH influences the ionization state of surface proteins that mediate adhesion. Acidic conditions can protonate carboxyl groups, weakening electrostatic bonds between cells. Alkaline environments increase deprotonation, strengthening those same interactions and promoting cohesion. The combined effect of flow, nutrients, and pH determines the overall stability of a biofilm. By adjusting these parameters in laboratory settings, researchers can observe distinct structural differences. Such experiments reveal how environmental cues orchestrate bacterial community architecture. Pause 0.8. Bacteria first adhere to surfaces through van der Waals forces and hydrophobic interactions. They then secrete extracellular polymeric substances, forming a mesh that glues cells together. Microcolonies grow into complex structures with channels for nutrient flow. Quorum sensing molecules signal dispersal by degrading the EPS matrix. Finally, shear stress, nutrients, and pH modulate biofilm formation and stability.